Welcome to the Maine Science Podcast. I'm Kate Dickerson. In today's episode, I spoke with Romain Madelaine, who is a new faculty member at MDI Biological Laboratory. Romain just recently moved from France to Maine, and his work in tissue regeneration using zebrafish is really interesting. Romain does a great job, I think, in explaining how basic research works and connecting the dots to why it's so important to science and to our understanding. I hope you enjoy our conversation. Romain, welcome to the Maine Science Podcast. It is a pleasure to have you with us. You are a new faculty member at MDI Biolabs, and some of our listeners will know what they do, and we'll get into that. But first, I would really like to know your background in science and how we were lucky enough to entice you to come to Maine by way of France.
I got interested in biology and genetics a long time ago. It was during high school, when I was able to understand that genes were able to make incredible things, like controlling the color of your hair or the color of your eyes. And I was, okay, this is amazing. And I would like to understand more about genetics and how we can control expression of genes and how we can have organs and whole body functioning. So I decided to go to the university in Toulouse, in France, to do my PhD in biology, in developmental biology. And I was working in the lab of the Dr. Patrick Boeder with the zebrafish. So as you probably know, zebrafish is a fantastic model organism for research. It's fantastic not only because it's small and cute, but also because it allows us to really do a lot of fantastic things like whole body imaging, fantastic because we can really manipulate them genetically to be able to study the function of genes. I fell in love with zebrafish during my PhD, and since I continue to work on this animal model since now about almost 15 years. So I did my PhD with Dr. Patrick Boeder, working on neurogenesis, developmental biology. And at this time, I was interested to understand the function of genes, of transcription factors, and how these transcription factors are able to control the expression of genes, to control the identity of the neurons. Because we know that we have billions of neurons in the brain, but a lot of them are different and are not making the same functions in the brain. So really, during my PhD, I studied how transcription factors can control the expression of different genes and how we can control the identity of neurons. So I was working in this lab for 4 years. And after I decided to move at Stanford to do a postdoc, still working with the zebrafish. But here, my question was a bit different. It was the lab of the Dr. Philippe Morin at Stanford. And here I wanted to understand how neurons can control different kind of behaviors. So not only the identity of the neurons, but also how neurons can interact all together to form functional neural circuits that can drive different kind of behaviors, complex behaviors like sleep, feeding, or pain response. So I spent few years working on this subject with the zebrafish, and here the zebrafish again is a really, really good model because we can do whole brain imaging, meaning that you probably know that the connection and the communication between neurons is by electricity, action potential to communicate and to drive a different kind of information. And we can follow this activity in the brain of the zebrafish with different kind of techniques like calcium brain imaging. So we were using this kind of techniques to understand the function of different kind of neurons and different kind of neural circuits in complex behavior like sleep, for example.
I know we have billions of neurons in our brain. How do you figure out even the basics of which ones to look at for the different parts that you're studying?
So we know that different kind of neurons will express different kind of neuropeptides or different kinds of neurotransmitters. So we basically determine the identity of one neuronal population by the neuropeptides that they can express, and we can manipulate the genome of the zebrafish, for example, to express some fluorescent protein in this specific population. So if we know that a specific population of neurons express a neuropeptide involved in sleep, we can look with microscopy, confocal microscopy, most of the time, we can look deep in the brain of the fish and see these neurons. And after, with different kind of imaging techniques like calcium imaging, with another type of manipulation, these neurons can express a calcium indicator that will allow us to look at the activity of the brain, meaning that if there is action potential, the calcium will be high and we'll be able to follow the activity.
And so you looked at sleep for your PhD work, right?
During my postdoc, my first postdoc at Stanford, yeah, in sleep and pain response mostly, yes.
And I know that MDI Biolab and you now are looking specifically at tissue regeneration. So I have two questions. How do you go from sleep to that? And then what got you interested in the tissue regeneration and muscular stuff and the brain aspect of that?
Yeah. So what you mentioned here is my two most recent projects that I will develop in my own lab at the MDIBL as a new investigator. So and how I moved from basic early neurogenesis during my PhD to study the function of neural circuits during my postdoc to regeneration of neurons or muscles is because I was not only working on sleep and associated behaviors during a postdoc, but I developed some other projects that I can tell you a bit about this project just after. And these projects led me to move to the regeneration part. And it's really something that I love. I think that it's amazing to see how the zebrafish is able to regenerate any part of the body, but most importantly, any part of the brain, of the spinal cord. So I was, okay, this is what I want to do next. And in the future, and about my long-term project is to understand what is the mechanisms of regeneration in zebrafish to maybe be able to apply our discoveries for regeneration in mammals and maybe one day in human. So how I decided to do this project is that at Stanford, I was collaborating with some great people and great labs. And one of the projects was to use the zebrafish to understand the function of mutations that we can find in a human DNA. So we know that there is some SNP mutation that we call single nucleotide polymorphism mutation. So it's one nucleotide mutation in the DNA. And most of the time, these mutations, or not most of the time, but sometimes, these mutations are associated to some kind of human diseases. It can be very, very broad. It can be diabetes. It can be degenerative diseases. It can be aging defects, many, many different things. But here, the goal of the project was to compare the sequence of the DNA, the conservation of the DNA between human and zebrafish to be able to say, oh, we have a strong conservation in this part of the DNA between two species that are very, very far, more than 450 million years of differences in the evolution between human and between zebrafish. So if we have a strong conservation in this part of the non-coding DNA, maybe it's because this mutation is not only associated to the disease, but maybe it's responsible of the disease. So we were working on that project for many years. And at the end, we were able to show that indeed, for some of these mutations, it was really responsible of the disease. And what we showed is that if you play with these mutations, so you have the mutations that can make you sick, or the mutations that make you behave normally and to be completely healthy. If we change the nucleotide, we were able to see difference in the expression of some genes and probably that these genes are responsible of the disease. So we identify different genes associated to human diseases. And one of them was a microRNA. So microRNA are pretty famous because more than 10 years ago now, a Nobel Prize was given to Greg Mello and Andrew Fire about the microRNA discovery, and all many people after was working on that, and we discovered that there is a lot of microRNAs and they can control the expression of many different genes. So very important for physiology and for diseases. And one of these microRNA is miR9, and I was working on miR9, and these genes we were able to show that it was responsible probably in human of the disease, because if we play with the mutation of the gene miR9 in zebrafish, we have some phenotype, some defects that were similar to what we expect to see in human with the disease.
It's amazing when you think about it, right? You said 50 million years ago, and we still these connections between this little tiny fish that you can see through and us. It's not obvious at all that that's going to happen. It's really amazing.
Yeah, it's true that there is a big distance in we think about the evolution, but in terms of conservation of genes, or in terms of conservation of function or development organs, it's very similar. So that's why it's really a good, still a very good model to understand human diseases.
So you said you fell in love with the zebrafish. I'm going back a little bit. Before that, were you trying to figure out how to research genes, or were you lucky enough to kind of find the zebrafish right away and dive into it?
In fact, no, I not right away, because before to come in Toulouse for my PhD, I was in Britain. I don't know if you know a town. It's quite a big city. It's Rennes, so in Britain, and I was here for my bachelor and my master degree. And at this time, I was not working with zebrafish. I was doing some research and rotation, working with the bacteria, and I love microbiology also. So it was really nice. But really, what I wanted to do is dev bio at this time, and it was not possible to do that in my university in Rennes. So I moved in Toulouse University, and here I decided right away to work with zebrafish, because Dr. Blader, he was my first mentor, and he was one of the first doing zebrafish in France. And it just convinced me that it was the best model to do a good biology. And since I can tell that I'm trying to go into doing the same and to explain the same things to my student. And I think that it's really a model when you start to work with it and to see everything that you can do. Really, it's hard to move and to work with something else. But I have to say that every model in biology has some strong advantages, probably also some weakness. But for zebrafish, I have to say that it's really a lot of advantage.
We need to make a zebrafish shirt for you so you can promote how great zebrafish are.
I'm sure that the MDIBL and probably University of Maine already know that for sure, because there is a lot of beautiful labs working with zebrafish already in Maine. So
it's an amazing organism. I was lucky enough a couple of years ago to tour a zebrafish lab and facility, and I was just blown away at not just the science that was happening there, but how much could be done in a relatively small space, and how vital these fish were to help us figure out kind of basic biology for everybody. It was really extraordinary.
Yeah, it's true that for basic biology, it's really great. And in terms of cost, of course, because we always need to take that in consideration. If you need to compare what you need to house mice or zebrafish, it shows that at the end the cost is not the same. So for basic research, understand molecular mechanisms of different biological processes, it's sure it's a great model. And we know that many of the things that we learn from development are involved after. Many of these mechanisms are involved in a different kind of diseases and pathologies like cancer or degenerative diseases. So yeah, basic research is really great for that. And zebrafish, since the last 10 years, 15 years, really became a very fantastic model for biomedical research also.
So you said you please correct me if I have this wrong. Your hope is that you figure out things in the zebrafish genetically, and that hopefully from there, you or someone else will be able to go to mammals and then eventually to humans. And I'm guilty of this, as many people are, right? The basic research is it's hard to understand because you don't see something right away, right? But if anything, what COVID I'm hoping has helped people realize is basic research sets us up, right? If we don't have basic research, we don't have mRNA vaccines, right? It really is that simple.
Basic research is before everything.
So just to give us a sense of how you think this would work, and I'm not going to hold you to it, but you figure out something in the zebrafish. How hard is it to go to mammals then after that? Like how hard is it in the lab? How hard is it time-wise? Like, what do you think? And maybe you can't answer this. I don't know.
I can answer probably. So yes, what I would like to do is to use the zebrafish to understand the mechanisms of neuronal regeneration, for example. And what we know is that zebrafish is a vertebrate and it's able to regenerate. So if we stab the spinal cord, the fish will not be able to move, but after basically 1 week, it will be able to move again. If we stab in the eye, it will lose the sense and the vision, but again, 1 week after, it will recover the function, visible function. So it's a same in the brain, different part of the brain. So really, we know that in mammals, some tissue like the liver, like muscles, are able to regenerate. If we do exercise, we will regenerate some part of our muscle tissue, but we are not able to regenerate the brain. Zebrafish can do that. So really, what I would like to do is to understand why and how it can do that, to be able to translate that in mammals. And what we know is that basically the same type of cells that are able to promote the neuronal regeneration in zebrafish, so what we call the neural stem cells or neural progenitors, are also the same type of cell with almost the same identity, are also present in mammals. In the eye, for example, that I study, these cells are called Muller glial cells, and we have these cells in zebrafish and in mammals. But in zebrafish, if we do the injury, these cells will proliferate, make new neurons, and restore visual function. If we do the same thing in mammals, we will have like the beginning of a proliferation, but it will stop, and these cells will undergo gliosis. So we have some break in mammals that prevent us to be able to regenerate following injury in the eye, for example. But it's probably the same kind of idea and concept for the whole nervous system. So really, I would like to be able to use new techniques like a single-cell RNA sequencing, like a CRISPR-Cas9 genome editing zebrafish to really be able to identify some genes activated after injury, probably involved in regeneration. And if we are able to show by classic gain, loss of function experiment that these genes are indeed important for regeneration, to test them in mammals. So a lot of labs are already doing that, and there is some beautiful results, but every lab tries to put a small piece of that, and we hope that at the end of the day, we will have a big breakthrough, maybe in 10 years or 15 years, maybe earlier if we are lucky. But really, we can test it in mice pretty quickly. I mean, I was collaborating when I was at Stanford with Jeffrey Goldberg, the chair of the ophthalmology department, and we started to test the function of some of these genes that I identified during my postdoc. And I believe that here, I will continue to work with him if necessary to test some genes, but we can also expect to have some beautiful collaboration. I know some labs at the JAX also doing this kind of thing. So it will be more than easy to just cross the road basically and to discuss and to say, hey, we have some good candidates in zebrafish. It will be nice to test it in mice and see if we can make new neurons.
So it sounds like you said we have these genes and it starts, but then it stops. do we have any idea why zebrafish developed it and mammals haven't? Like, was it more like other parts of our body became more important genetically and evolutionarily, and our genes just decided that wasn't important enough? And again, maybe you don't know, but I'm kind of fascinated by this idea that we've got all the basic raw material there, but somehow it doesn't work for us right now.
Yeah, that's correct. So basically, yes, we have the good cells, good neural stem cells in our body, in our brain, to do that. Our genome is able to express, or should be at least able to express, the good genes, but it doesn't work properly. So what we start to understand, and it's really more and more common now to see that this is basically true for many kind of diseases and during aging and many different things, but it's mostly about the chromatin and the regulation of the accessibility, to make it simple, of the genes to be activated or not. Probably that some genes that should be activated are not accessible, because what we know is that in mice, for example, early during development, mice are able to regenerate. It's not very good, not like a zebrafish, but still it's possible. But in adults, it's not possible anymore. So something happened, and probably that some genes are locked down and are not able to do the job. So it really, everything is about control of gene expression.
It feels like we're in this really remarkable age of biology and genetics, that everything that you're talking about sounds almost like science fiction, because I think people have been thinking about it for a long time, but now we're at this point where we can really do it. So it leads to all sorts of really interesting questions about the science, but also kind of the ethics of how do we figure out what to turn on, what to turn off, what are the repercussions, what are unintended consequences, some of which it seems to me you might be able to find out in your research, right? So if you're able to turn on and turn off the genes in the zebrafish, can you then see what happens around it, and that helps you guide the next steps? Does that make sense?
Yeah, completely. It's exactly what we want to do, is that we at every step we need to understand what is going on. So how we activate the stem cell to proliferate, what is controlling proliferation, and what will control the differentiation of the progenitor in neurons, and not any neurons, because what is also very important, I told you at the beginning, but I was interested in the identity of neurons. This is critical. We just want to make any type of neurons. If I need a retinal ganglion cells to see something, I don't want to make a new photoreceptor. So yes, we need to be able to check at every step what is happening about the expression of genes. So now we can do that. I will not say that it's easy, but we have new tools, and single cell sequencing is a beautiful tool to understand what is happening at every step of the differentiation. And what also is important is that, like I told you, there is many labs working on that, and basically we have all different kind of candidates. I have few candidates myself that I will test first. I hope to discover more and to be able to test them in the future, but maybe I will be able to make more photoreceptors, and maybe another candidate in another lab will help to make any other type of neuron of interest. So we will need to just work every day to test hypotheses and to see what happened.
So maybe you could, I realize we're talking at length about zebrafish and a zebrafish lab, and you and I know what it looks like because you work in one and I've been able to tour it, but maybe you could explain what it looks like to someone who hasn't done a lot of biology. Would you mind doing that?
What, a zebrafish, or to work with a zebrafish?
What does a zebrafish lab look like? Like what if I walked into your lab, what am I going to see? But also, I realize this is very basic, but how do you do the experiments?
So the most important in a zebrafish lab is always the fish facility or the fish room. So I am lucky enough to I have always been in a lab, an institute with a beautiful fish room and a lot of space. So basically, it's a lot of tanks So small tanks, big tanks with water and a fish in it. We can have thousands of fish in one fish facility. So we have a beautiful fish room at the MDIBL, and I'm lucky to be able to bring all my fish and to have them very soon. And really, it's just a room full of fish. So it's always really nice for students, because when they come, they always love to see zebrafish, and I think that for us it's really good, because it really allows us to just catch some of the best students to come to work with us, because we love to see that. So the most important is the fish room, and after it's a lot of microscopy. Zebrafish became really famous because larvae are transparent. So really, what is really important in zebrafish research, most of the time, it's microscopy. So here we have some small binocular microscope to just look at them. It's a microscope that we can see basically everywhere, just to have a close-up and to look at things, and to look at the biology and the beauty of what we are doing. And also what is really important with the zebrafish is the genetics. I told you that we can do that. So here we do what we call microinjection to manipulate the expression of a gene. So to make transgenics or to make mutants. We can play now with that, and we do microinjection. So basically what we do is to inject with a small needle some DNA or RNA, or DNA and RNA in the eggs of the fish. So it's one cell, just at the beginning of the development. It's only one cell, and we inject with a needle in this cell, and we try to modify the genome to create new genetic tools. So this is, at least for me, what I do the most, is to create new tools to try to understand the biology.
And what do you hope to do at the lab start? Like, do you have a couple of projects as soon as you are able to hit the ground running, the few things that you're going to work on and then build up? I know you said you're working along with collaborations with other labs, but what are the very specific things you want to start with and then moving on from?
So I will continue to work on the two projects that I already started. The first one is the one that I started about neuronal regeneration that we already discussed before. So I will continue that project. I will test some candidates that I have for their capacity to reprogram endogenous neural stem cells. So here there is no problem of ethics, of transplantation of cells, or everything. We just want to use the cells already in zebrafish and maybe in mice or human to make new cells, new neurons. So this is the first project. And the second project is about muscle regeneration. So this is a much more recent project that I started only 2 years ago. So it's really, I will not say that it's not yet mature, but we really are working on it. But here the idea is basically the same, is to be able to understand why with aging, the muscle is not able to regenerate properly and efficiently. Because contrary to the brain, humans are able to regenerate muscle tissue. If we have atrophy because we had some kind of injury, we are able to do exercise and to regenerate muscles. But this is not true for most people when you are older. And something happened. It's aging, so probably modification of chromatin and gene expression and all these things. But what I would like to do here is to identify some of these factors that makes the muscle tissue not able to regenerate, to be able to promote regeneration of the muscle tissue. So here again, using the zebrafish to test candidates, to test genes, and after maybe to apply it to mammals. And here the main disease associated with aging and muscle is sarcopenia, that you probably know. So this is my second project. So it's all about cellular regeneration, one part in the brain, one part in the muscle tissue.
Those are two very different areas. How much crossover is there, or are they fairly separate?
They are not, it looks obviously different, but what is really different is just the tissue. Basically, the biology, I will not say that the same factors are involved, but most of the time we know that not the same transcription factor, maybe not exactly the same signaling pathway, but we know that most of the signaling pathways that we know to be important for development are also important during pathologies and are probably involved in the defects in weight generation. So we know that if we play just with some pathway, notch pathway, so maybe I'm becoming a little bit too specific, but notch pathway, Wnt pathway, all are involved in the control, in the balance of quiescence, activation, proliferation. So it's not so different. And after, the techniques to manipulate genes, to study the effect of our genetic manipulation using microscopy or single cell sequencing is basically the same kind of techniques.
I personally, from a science point of view, am really glad that you'll be joining us. And I'm fascinated by the idea that you're working on what at first glance looks like two very different parts of the body and different species, and that they're just so connected. I think it's really interesting and really cool stuff. Thank you for your work. Safe travels and safe moving and all of that. And I can't wait to meet you in person and hear more about your work as it goes on.
Thank you. It was very nice.
The Maine Science Podcast has received support from the Maine Technology Institute and is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is produced and edited by me, Kate Dickerson. I received production support from Maranda Bouchard. The Discover Maine theme was composed and performed by Nick Parker.